Dear students of 12th class, today in your botany we will discuss the incomplete dominance. Till now we have discussed in detail the Mendel's laws. Out of his four, we have given the explanations to three of his laws. And now today we will take up a slightly different, that is slightly away from the Mendelian in this research, that is the incomplete dominance. Incomplete dominance. Mendel carried out, as you know, his experiments with cardon pea as his experimental plant, and when such hybridization experiments were repeated with the characters or traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the parents and was in between the two. Hopefully you understand this. As in case of the Mendelian crosses which were made with the pea plant, all the traits that he studied, the F1 produced the generation that had only one type of the phenotype. One phenotypic character was that evident there, only one out of the two. For example, in case of the height, Mendel showed in his this hybridization that all the F1 plants were tall. That means the tall trait was dominant over the dwarf trait, and here it was a slightly different trend away from the Mendelian, that is the F1 had a phenotype that did not resemble either of the two parents, and it was in between the two. That means the phenotype was in between the two, it resembled neither of the parents. Here, in case of the snapdragon, of which the example has been quoted, the flower color was taken as red and white, with the red being dominant over the white colored flower, and the result was that the inheritance of the flower color in the dog flower, it is snapdragon or antennatum species. It is a good example to understand the incomplete dominance, because when a cross was made between the true breeding red flowered, denoted by capital R, and a true breeding white flowered, denoted by small r, the F1 hybrid, that is capital R, small r, were all pink. They were all pink. That means the F1 generation, in case of Mendel, it should have resembled either of the parents, but here it resembled none of the parents, and all the plants were bearing the flowers that were pink in color, which was resembling neither of the parents, because the parents had red and white, whereas in the F1 generation all plants were with the flowers having a pink color. So when F1 was self-pollinated, the F2 resulted in the following ratio, as 1 pure red, that is represented by capital R is to 2, and that is a hybrid, that was pink, and 1 represented by small r white. 1:2:1 ratio, 1 red, 2 pink, and 1 pure white. Here the genotypic ratios were exactly as would expect in any Mendelian monohybrid cross, but the phenotypic ratio had changed from the 3:1 dominant recessive ratio. What happened was that the capital R was not completely dominant over small r. That means the red flower, red character of the flower was not completely dominant over the white color, and this made it possible to distinguish the hybrid, that is capital R small r, as pink from red and white. Hopefully you understand this all. Now we have been dealing with these classes from the previous 3 or 4 classes. This here you can see, this is the parent generation at the top with the red color, that is represented by capital R, and white color by small r, and the gametes with half, that is R, capital R representing and the red, and the small r representing the white. And here through the Punnett square you can see the ratio of 1:2:1 with 1 red, 2 pink and 1 white, and the genotypic ratio of 1:2:1. Phenotypic ratio 1 red, 2 pink and 1 white, and the genotypic ratio 1 red, 2 pink, hybrid pink and 1 pure white. This is the monohybrid cross in Snapdragon that shows the incomplete dominance. And now the dominance, every gene contains the information to express a particular trait. This is important. Every gene contains the information to express a particular trait. In a diploid organism, there are 2 copies of each gene. That means the allelic pair. That's a pair of alleles. Now these 2 alleles need not always be identical as in a heterozygote. One of them may be different from the other due to some changes that it has undergone, which modifies the information that a particular allele contains. That means that changes are possible. That is the reason why the Mendelian that crosses, it has a deviation from the Mendelian monohybrid cross that he performed with the pea plants, and here it showed the incomplete dominance. Thanks, have a good